So here we continue with the plant cell with a few different. First, we will discuss the different things which are present in the plant cell. Number one is plasmodesmata. Here is a plasmodesmata. Here you can see it. This one is the plasmid. So what is plasmid? Actually, it is a gap in or a pore, you can say, in the cell wall, which connect one plant cell to the second or adjacent plant cell. So this one is the cell wall of the adjacent plant cell. Here you can see that this is the cell wall of the second plant cell. But here it is discontinued here, and then you can see here. Okay, so here we can look about. So this one is the hole through which the cytoplasmic content can move from one cell to the other cell. And so this is the connection between the cytoplasm. So simple divisions, the nutrients or the materials can be exchanged between these two cells depending on the concentration gradient. So number one difference, plasmodesmata. You can say plasmodesmata or plasmodesma. Both way you can write. Okay, cell wall is most commonly the difference between animal and plant cell is here, and then we are not finding anything in this list here. And then Golgi apparatus might be some missing in some of plant cells, maybe, but most of the plant cells contain Golgi apparatus. Here is the chloroplast. Chloroplast is the second largest organelle that is present in the plant cell, and inside there is a grana. Grana are the structures which can trap the light during photosynthesis, light-dependent reactions. So this one is the grana. Here is the complete structure of the chloroplast, in which grana are interconnected also. Here, chloroplast again, and it has a double membrane. Chloroplast has double membrane. It has also 70s ribosomes and circular DNA. The things which are present in the mitochondria, same things are present in the chloroplast. 70s ribosome. In the cytoplasm, there is a ADS ribosome. Most of them are attached with the rough endoplasmic reticulum, which is here, rough endoplasmic reticulum here, rough endoplasmic, which is near the nucleus. So this one is the nucleus. You can see there are nuclear pores also, and there is heterochromatin and euchromatin both, as I told you earlier in the animal cell structure. So this linear chromosomal DNA is euchromosome, you can say, or heterochromosome is lies just side of the inner side of the equilateral. So this free ribosome has a size of ATS. ATS means that it has 2 units. So SS means subsidiary units are there. So actually it is 40 and 60 S. 40 and 60 S. So 40 and 60 S mean that it is total, it should be 100, but it is 80. So 60 S plus 40 S should be 100, but it is becoming 80. 80 S. So due to the overlapping regions of the ribosome, if I can draw a ribosome here, as it is at 25 nanometer, but I am making much larger. Yeah, so this is the smaller subunit here. This one is the large subunit, and this one is the small subunit, and both are overlapping, so their total size is 80S, not 100S. Anyway, on the smaller subunit of ribosome, messenger RNA there. And then this you can study in later lectures of the chapter number 6, protein synthesis. So now next one is the most important, very much large vacuole is here. A tonoblast is the membrane of the vacuole, and one large permanent vacuole is present. This is one of the difference between the animal vacuoles and plant cell vacuoles. Actually, there are many difference between the vacuoles. One is large, and the animals has small vacuoles. Number two is it is permanent. So permanent mean that animal's vacuole has the property that it form, and then uh, in some of the animal cells it break down, because they are doing the function of excretion in some kind of the contractile vacuole, one of the type of vacuole. There are many types of vacuole is there in animal cell. So here in plant cell there is no other type. So it is only one type, that is a permanent large and central vacuole, you can see. So it cannot be break down throughout the life of a plant cell. So what does the vacuole contain? It contains the sap. What does the sap contain? Sap contains water, mineral salts, glucose, and amino acids. So this one is the sap. The vacuole sap or cell sap, whatever you choose the word, is good. So here the missing thing is the centriole. Centriole is not here near the nucleus, as we have told in animal cell. The centriole is cylindrical structure is present in plant cell. Here is the comparison between two types of cells. One is prokaryotes. Prokaryotes is here, and then eukaryotes is here. So prokaryotes are mostly bacteria. They are lacking with the nucleus, and eukaryotes are animal cells and plant cells, and fungi cells are as well. So number one comparison we have done, that type of organism in which the prokaryotic cells are present, that is bacteria, and here is animal, fungi and animal. And the cell size is 0.5 to 1 micrometer. It is not 10, it is 1 micrometer, 1.0, 1.0, which is the size of smallest mitochondria as well. Or some vesicles also have the same size of 0.5 micrometer. But the eukaryotic cells may be 100 times larger than this one. So it can be 10 micrometer, or maximum is 100 micrometer. So on average, we can say here is that it is on average is 40 micrometer. Most of the eukaryotic cells are there. 20 micrometer are most common, and average size is 40 micrometer. Okay, and then it can be only unicellular. Maybe they are in the form of colonies, but they remain as individual in a unicellular organisms. And they may be unicellular and multicellular in case of eukaryotic cells. So mainly they are multicellular, except Prokaryotes. So what are the Prokaryotes? Plasmodium, in all levels you have studied that it causes malaria. Plasmodium is Prokaryotes. That is not bacteria. So it is a unicellular pathogens which have a nucleus. Also they are known as Prokaryotes. So Prokaryotes, many of which have unicellular structure. Algae as well. Algae also are eukaryotic, and some of the algae has unicellular structure. So evolutionary origin is 3.5 billion years, and here is 1.2 billion years. So this show evolutionary trend in between the prokaryotes and eukaryotes. So how the cell divides of the prokaryotes is by binary fissions, because they have no nucleus. In mitosis, we are saying nuclear division, so because they are containing nucleus eukaryotes. So that's why we are saying mitosis or meiosis, because the presence of nucleus. So spindles are formed here, so there is no spindle formation. There is no spindle formation in prokaryotes. In eukaryotes, spindles are formed here due to the presence of centriole. Then genetic material, DNA is circular. Circular means that which has no end. So this one is a circular DNA, so it has no ends here. So it's circular DNA or loop DNA, you can say. But it has linear mean that which I've told you that it is eukaryotic. So it has two ends. So it is a thread-like structure. So they are linear DNA in the nucleus. No doubt they have also a circular DNA as well in the mitochondria and chloroplast in case of eukaryotic cells. So it means that it has both DNA, both type of DNA, circular DNA and linear DNA as well. But the nucleus contain only linear DNA. So the DNA in prokaryotes is not associated with any type of protein or RNA, so to form chromosomes. So not associated with proteins, prokaryotes. And DNA is associated with proteins. And can you name the protein which is present in the DNA? That is histone. Histone protein. So it stabilizes the DNA linear structure so that it's not tangled. Next one is the protein synthesis. Protein synthesis consists mainly of the 70S ribosome on which the protein synthesis are taking place. There is no rough endoplasmic reticulum because there is no membrane-bounded organelles. So there is no organelles which has the membrane. A few organelles may be present which do not have envelopes or double membrane as well. So ribosomes may be attached to the endoplasmic reticulum. So except ribosome, which in some books written that it has a membrane, but we know that it is present in prokaryotes 70S ribosome. And in case of eukaryotes, there is a 80S ribosomes here, 80S. No doubt the 70S ribosomes are also present in case of this 70S ribosome in case of mitochondria and chloroplast, within mitochondria and chloroplast, not in the cytoplasm, but no doubt they are present in the eukaryotes. So 70S and 80S, both ribosomes are here. So here are many organelles in comparison to the prokaryotes. A few organelles mean that only ribosomes. And here many organelles are there, mitochondria or Golgi apparatus and lysosomes or vesicles and so many other. So all the organelles have the membrane structure. So cell wall in bacteria, the cell wall is made up of peptidoglycan or murine. Murine, you can also see this murine, murine, murine or peptidoglycan. These are the complex carbohydrate structures associated with some peptides. So here in case of eukaryotes, the cell wall is mostly of cellulose in case of plant cell. And in case of fungi, it is made up of chitin. Chitin in case of fungi, yeast you can say. So there are two types of components here now in eukaryotes. The cell wall is made up of cellulose in plants. Green plants have the cellulose, which is a polysaccharide, and the chitin is also a complex carbohydrate, and the cell wall of fungi is made up of that. Now, one of the most important thing is flagella. Flagella is simple lacking microtubules. Microtubules are the proteinous strands, so they are not in the prokaryotes. Some of the bacteria have flagellum or flagella to move in the fluid mediums. So here is complex 9 microtubules in the form of duplet. Duplet mean that loop, not 3 here. You can see here, there are 2 microtubules, cylindrical structure, top to bottom move. So that is a cylindrical structure. Top to bottom it can move to form a hollow cylindrical structure that is in pairs, and there is 9 pairs altogether. 9 pairs, you can say 9 pairs are there. So second pair, then third pair, then likewise fourth and fifth, and then 9 pairs. 9 pairs of duplicate microtubules are here. So this is one of the differences that this flagella of prokaryotes do not have the microtubules. They have the microtubules, and their structure is also mentioned here, that it is arranged in the form of pairs, and there are 9 pairs. The other difference is that the flagellum is not enclosed in the cell membrane. So it is naked proteinous structure and not enclosed in the membrane. It has a 20 nanometer thickness in diameter, where it has surrounded by the cell membrane, because this flagella is originating, for example, if this is a eukaryotic cell, and the flagella just originate from here inside. From inside it is very, so it has the basal colony here. From here it originate and then spread out in the surrounding. So along with cell membrane is there, along the side of this, the cell membrane is there, so its thickness increases here. So its thickness increases, so then we can say that it is 200 nanometer in the diameter. Its width is 200 nanometer, like this, 200 nanometer diameter. So now the three difference we have: one is the microtubules difference, number two is the enclosing membrane or not enclosing membrane, and third one is the origin point. Origin point in case of the this one bacteria, this origin is just from the outer surface and there is no membrane. It is not down inside the cytoplasm. And the four differences in diameter, 20 nanometer and 200 nanometer. So this is edition in 2023 syllabus of CI. So mostly they will ask questions from here after that. So respiration, this mesosome structure is now out of syllabus after 2023. Mesosome is the infoldings of the membrane. So infolding of membrane mean that here is the membrane. So infolding membrane is this structure. So this structure, which is infolding of the membrane here, may contain some of the pigments here in case of photosynthesis, and here in case of the respiration, they are carrying out production of ATP. They are present in the bacteria, infolding of the membrane, mesosomes, but now they are not in the syllabus. Here is the mitochondria, a complete structure, which has a complex structure also. So it is the second largest organism in the animal cell, which has the diameter of 0.5 micrometer to the 5 micrometer, depending on the shape and size of the mitochondria. So ATP production is over there during aerobic respiration. So in photosynthesis, there is no chloroplast, as I mentioned you, that mesosomes is the infoldings of the membrane. So they contain pigments. Instead, a separate structure of chloroplast, which is present in the eukaryotes. So third thing is also related to mesosome nitrogen fixation. So these infoldings membranes also contain some of the pigments or material which is carrying out nitrogen fixation as well. So these 3 points, respiration, photosynthesis, and nitrogen fixation, is related to mesosomes. So this is all about the comparison between the prokaryotes and eukaryotes. Any question you can ask at the comments. Thank you.